Okay, good afternoon, everybody, and welcome back to another of our recorded sessions. Today, we're going to be discussing animal diversity, preparatory to quite a lengthy discussion about the animal kingdom. And first of all, we're going to learn that there are certain things which make an animal, which distinguish it from, for example, the other predominantly multicellular groups, the fungi and the plants. So what makes us animals is the fact that we are multicellular, we are heterotrophic, obviously we're eukaryotic, and in particular that we have tissues which develop from early embryonic layers. And unlike the plants, can manufacture in their own organic material, we are required, because we are heterotrophic, we are required to use already constructed biomolecules as a source of elements, of molecules, etc., but also as a source of energy. Animals have no ability to resort to autotrophy, unless, and there are some specialized animals which have symbiotic relationships with algae and things like that. But inherently in themselves, they are not able to be autotrophic. The second distinguishing thing is that unlike the fungi, which are able to release enzymes and then absorb material from the outside, we actually have to absorb all our food internally. And we have to eat food. We have to digest it usually in a complex gut and reduce it to molecules which we can then absorb and utilize. So we are truly heterotrophic. The second thing which distinguishes the animals is the fact that around our cells, we have no cell wall. Both the plants and the fungi and the prokaryotes as well are surrounded by a thick cell wall. That is usually an osmotic protection, provides some other protection as well, and some structural support, etc., etc. Animals have none of that. Instead, cell membranes of different cells can be in close proximity to one another. And in fact, there are special mechanisms to tie cell membranes very closely together and allow cells to communicate with one another in all sorts of ways. Animals are multicellular, and that multicellularity has all sorts of implications. The close contact of cells in this way means that cells can begin to coordinate with one another. And they are able to form specialized groupings, which are tissues, and they're able to take tissues and put tissues together and form specialized organs. So this is a huge advantage, and it enables animals to develop organs which are highly specialized for particular functions, and also to have very specialized tissues as well. So in the animals, there are two types of tissue in particular, which are defining characteristics for the animals, and these are nervous tissue and muscle tissue. And I'll mention to you that even in the most primitive of all, there are the precursors for these tissues. Another distinguishing feature of the animals is that our predominant means of reproduction is sexual. There are animals that have asexual reproduction, that is true, but all animals have a sexual reproductive stage, which is of particular importance. And a reflection of that is the fact that the diploid stage dominates the animal life cycle. You remember in the fungi that the haploid stage dominated the life cycle. In the protists, you may have alternation of diploid and haploid, but in animals, any haploid stage, any haploid cells are usually very short-lived. It's a very short stage when the gametes are formed preparatory to again allowing fertilization to form a diploid zygote. So The sperm and egg cells are produced by meiosis, but that is a purely process of the phenomenon of reproduction, and those do not persist as life stage. Animal zygotes, once fertilization has occurred, they undergo a complex system of cleavage, of division of the cells. Now we're going to learn that there are actually two predominant means whereby cleavage occurs. But the results of cleavage is that a hollow ball of cells is produced, which is called the blastula. And in the hollow cavity in the middle, you're going to have to learn to follow these cavities and remember what their names are, because their genesis and their fate is very important. The cavity in the blastula is called blastocele, and the blastula undergoes a process whereby the blastula invaginates. It folds in on itself like this, and that is that process that forms the embryonic tissues. At its simplest, as we'll hear, at its simplest, 2 layers of tissue. But an elaboration of that is that 3 layers of tissue are actually eventually formed as we move through the evolution, through the phylogeny. So let's first of all just get you used to some of the names. Remember, this is the blastocele. It's the hollow inside the blastula. When this invagination takes place, another hollow is being formed here. And that there is called the archenteron. And it has an opening, and that is the blastopore. When it is simply 2 layers of tissue, the outer layer is called the ectoderm. And the inner layer that is formed from this fold here, that is called the endoderm. So that ecto means outside, endo means inside. Derm is just a layer of tissue. So these are the primitive embryonic tissues. This process of gastrulation is very complex, and it's very, very important. Some embryologists will tell you there are 4 key events in life of an animal. And the first is fertilization. The second is gastrulation. The third is puberty. And the fourth is death. And there's only one that has a certain outcome, and that is death. Gastrulation can go wrong. It is very, very complex, and the process of gastrulation sometimes, quite often, actually fails. And if it fails, the embryo is rejected and does not proceed, hopefully does not proceed anymore. So all of these very complex processes are governed by sets of developmental genes. I'm only going to mention the name of the gene, not describe to you its the functions. But in the early stages of development, a set of genes called the Hox genes regulate development. And they regulate development largely by monitoring the position of cells relative to one another. So they can tell where in this gastrula, where the gastrula is the result of gastrulation, they can tell where those particular cells are and what, because of their position, what their fate should be, and regulate that fate. The Hox genes control the expression of all sorts of other genes. So they control expression of things to turn cells into muscle, to turn cells into bone, etc., etc. So remember, please, that we did discuss the history of life on Earth. Please read that chapter carefully, because we had to skip through it, and there's a lot of information there that you actually need to know. But we remember that what we see around us now is enormous diversity in the animal kingdom, and we've identified about 1.3 million species. But there are far more than that that are completely unidentified, particularly in certain groups like the insects and things like that. Our molecular studies, together with studies of fossils and dating of sediments, et cetera, suggest that the common ancestor to the animals probably arose about 770 million years ago. So you'll realize that a long period had preceded that. And that period was largely dominated by the prokaryotes. Once the animal ancestors evolved, diversity developed first relatively slowly and then suddenly very, very rapidly. So when we look at the fossil record, the very first truly animal fossils that we see are here in about 560 million years ago. And these are the Ediacarans. Look back, I mentioned the Ediacarans before. Within the Ediacaran group, lie the roots of some animal groups which are still extant today. The root we see in the late Ediacaran, we see what look like ancestral forms, that may be ancestral forms of mollusks, annelids, various things like that. At this point here, the animal fauna is dominated by appears to be dominated by soft-bodied forms. So we have very poor record because those don't fossilize very easily. The fossils are extremely rare. But here, about 535 million years ago, there is a sudden explosion of hard-bodied forms and of many, many diverse animal types. And this we refer to as the Cambrian Explosion. The first early terrestrial vertebrates came up onto land about 365 million years ago, emerging from aquatic species. This here is called the Paleozoic, and that means the era of ancient life. The Mesozoic here is the one era which everybody seems to know about, because this is the time of the dinosaurs, of the flowering of the reptiles, and the dominance of this one particular reptile group, which were the dinosaurs. They died out about 66 million years ago, and all of their ecological niches were filled by the mammals, which had actually evolved all the way back here, but had remained a relatively obscure group because of the dominance of the dinosaurs. So when we go through all of that, what we realize is that there is, in fact, a common ancestor to all of the succeeding diversity. And we believe that that common ancestor was, in fact, a flagellate. And this is what it looked like. It's called a choanoflagellate, and I mentioned it in lecture. And here's its flagellum there. And it has this little crown around the flagella there, which it uses to filter food out of the water column. These still exist. They can be found commonly in marine systems. And there are a couple of things about them which are really interesting. First of all, they do form colonies, which may be a step towards multicellularity. But they are off on their own, in their own clade, and we would regard them as being a basal group from something very similar. We share a common ancestry with them from something probably very similar, which led to the animals. And our very simplest animal group are the sponges. And in the sponges, we see whole tissues made up of these choanoflagellate-like cells, which we call choanocytes. So that we regard the sponges as being a basal group in the animals. So these are not animals. These, in the old days, would have been considered protists. The true animals are those which are multicellular. But in the very simplest groups here, in fact, in the sponges, there are no true tissues. And so we put them off as a basal group. What is really interesting when we look at these choanocytes, these living choanoflagellates, and at our own tissues, and we look at it molecularly, we discover that they share DNA similarities, homology for a start, but they share homology also in some of the proteins which allow tissues to begin to form, and these are called the cadherins. And cadherin proteins are actually present in the choanoflagellates as well as in the animals. So I have set a piece of homework for you, please. And this is, I'm not going to show them now, but there are 6 slides here going through all of the geological record, the major characteristics of each of the eras, etc., etc. I would like you, please, to do a summary of those 3 printed pages. You may, if you want, include diagrams. That is absolutely fine, but you're not to take up space in the 3 pages with the diagrams. In other words, there must be a total of 3 printed pages. Okay, and that is due February the 14th. February the 14th, by the way, is your first test. So make sure that it's done before then and hand it in at that class. Okay, so when we look, now we're going to have to start to make sense of all of this diversity and try to decide how is it that the different animal groups are actually related to one another, if they are related. We believe they are because they share a common ancestor. And there are various things which we use as clues to help us work it all out. The first is that there are some different aspects of what we refer to as body plan, the way in which animals are in fact constructed. And many of these body plans have been conserved. They have existed, they were established very early on and have been elaborated on, but the basic body plan can still be discerned. And different groups, animal groups, have different body plans. And those body plans, the fact that they share those body plans is probably a good clue that they are closely related, that they're more closely related than they are to other groups with different body plans. So what we look at are some of these characteristics. First of all, symmetry, which I'll describe in a minute. The second are tissues. What types of tissues do they have, embryonic and in the adult form? Second of all, many animals have body cavities, as I'll describe to you. But those body cavities have different origins and different purposes. And the really important feature is the early embryonic development, how division in that zygote takes place and what the fate of those cells is. So all of these things are of importance, and I'll explain each absolutely clearly, I hope. The first, we're going to go to our basal group, our basal sister group, which are the sponges. Now, in actual fact, some zoologists would consider sponges as not animals at all, that they're in a gray zone between the protists, what used to be called the protists, and the animals. The reason for that is they have no true tissues. But when we look at them, we see, first of all, there is no true symmetry to although they may be round and they may be whatever, it's not true symmetry as I'll describe in a minute. They may be symmetrical, but it's a kind of an artifact of their existence. The truly radially symmetrical animals are things like the sea anemones, Hydra, those sorts of things. That is what we refer to as radial symmetry. And then the last kind of symmetry is bilateral symmetry. That is our symmetry. It was identical, not identical, but very similar from side to side. The body plan is bilaterally symmetrical. This is often a reflection of the lifestyle of the animal itself. These a lack of any symmetry at all, and radial symmetry are both of them associated with sessile animals, with animals that are fixed to the bottom, which do not move very much relative to the bottom. They usually do not have any rapid locomotion. Some of the radially symmetrical may be suspended in the water. There are tremendous disadvantages to this lack of symmetry. Bilaterally symmetrical animals typically are fast moving and also have a central nervous system. And the problem here are the problems. Okay, first of all, these that lack symmetry, any symmetry at all, it's very difficult for their tissues to organize into perform particular function. It can be done, but it does not appear to be very common. In the radially symmetrical organisms, they are receiving information from all sides equally. They have no front, they have no back. It's very difficult, for example, for radially symmetrical organisms to develop a nervous system because they require equal sensory input from all around. And as a result, they tend to have more simple nervous systems, which are often nest-like and equal all the way around. The bilaterally symmetrical organisms have a huge advantage because they have a front and they have a back. And where do they need the most information? Not in all the information, but most of the information is most useful coming in from the front. And they tend to develop elaborate sensory organs at the front, together with an elaboration of the nervous system. And the nervous system itself is asymmetrical. So they have a central nervous system, and they have processing centers along that central nervous system. It appears that bilateral symmetry is much more advantageous body plan that evolution can actually work on a lot more than can on radial symmetry. So here are some of the radially symmetrical organisms, and these are probably familiar to you a little bit. I think you may have seen them in lab. You probably will see in lab if you have not already. So the body in the plan is that they're radially symmetrical. In other words, they are symmetrical around a central axis. All you have to do is look at them from the top, and you realize that it's a circle with equal organs coming off all around. And there's no left, right, etc. But that's not to say that they're just a ball with things sticking out. They may be asymmetrical from top to bottom, but they are radially symmetrical in their presentation to their environment. So those are the typical radially symmetrical, and you'll immediately realize these are things like jellies, Hydra, sea anemones, those sorts of things. We'll see that when we talk about it, the invertebrates. Bilaterally symmetrical organisms are the vast majority of the animal kingdom. And in bilaterally symmetrical organisms, they are organized across two axes of orientation. And if you take a slice down the middle, you roughly describe that side as a mirror image of that side. Not entirely, of course, because even invertebrates are handed. Often one claw may be bigger than the other, but the basic plan is reflected. This immediately does something else. It gives the bilaterally symmetrical organism a front, and it gives it a back. It gives it a right, and it gives it a left, and it gives it a top, and it gives it a bottom. The underneath, the bottom, is called the ventral side. The top is called the dorsal side. For us, our ventral side is our tummy area, and the dorsal is the spine side. So this asymmetry, this bilateral symmetry, enables specialization of the sensory equipment and the specialization of all sorts of other things. It's a very advantageous body plan. And the most important of all of these specializations I've already mentioned is the process of cephalization. It's the concentration of sensory apparatus here at the head. And together with that, the development and elaboration of the nervous system initially to service those sensory organs. So let's have a discussion now about the tissues. I'm just trying to move my face out of the way. So the tissues of an animal develop in the embryonic stages, obviously, and they develop through this complex process of cell division, which first of all leads to the production of the blastula. Here's the blastula here. Inside, what is this called? The blastocele. Okay, it's the hollow blastocele. The process of gastrulation is the process of invagination of the blastula. And in doing so, what it's doing two things. First of all, it's creating a hollow on the inside. And second of all, it's creating a hole into there, into the inside. Here's the blastocele. Here's the gastrula. Once gastrulation is, once that invagination has taken place. And this hollow here is the arc enteron. Sorry, my computer is collapsing. This hollow in here is the arc enteron. And this hole here is called the blastopore. And make sure you understand the position and the origins of the blastopore, because it becomes very important. Here is the ectoderm here, and here is the endoderm. At its simplest, embryonic, that is how the embryonic tissues are formed. And there is one particular group that we're going to talk about where it stops right there. And the resulting animals have just two body layers. They have tissue around the outside, the ectoderm, and they have tissue on the inside here, which is the endoderm. And they have a blind gut. They do not, that only has one opening. And those would be the things like jellies and Hydra and those sorts of animals. They only have the ectoderm and the endoderm. We're going to learn that most other animals fill this cavity here. And they fill this cavity in there with tissue, which we call the mesoderm, the tissue which lies in the middle. And that has all sorts of consequences. This endoderm, whether there's just one opening or two, doesn't matter. This endoderm gives rise to the gut. In some animals, it's a blind gut, and we call that an incomplete gut. In others, there's an anus develops, and the gut is called complete. So the animals with two layers are called diploblastic animals. And Hydra is a typical example. Here's a cross-section of Hydra. And you can see here the ectoderm. And then here is the endoderm. And in between, in between the two, there is no other tissue layer. There may be a jelly-like layer. That's what makes the jelly of the jellies. And that's called the mesoglia. In the triploblastic animals, there are three tissue layers. And here's the simplest example here. This is planaria, which is a flatworm. And they have three tissue layers. Here's the ectoderm there still. Here's the endoderm, also forming the gut. But in between, there is a tissue called the mesoderm. And from the mesoderm, develop all sorts of other organs. Even in these very primitive triploblastic organisms like the planaria, there are all sorts of complex reproductive structures and all sorts of things. In primitive kidneys and lots of organs can develop from this mesoderm along with muscle. So that is the mesoderm in there. Now, the organism I've just told you about, the flatworm, the planarian, they have no body cavity apart from the gut cavity itself. And in these simple triploblastic animals, the gut is still incomplete. There's only one opening, and that functions as both mouth and anus. So they have to eat, and they spit out waste through the same opening. Here's if we cut across there, here is the structure, ectoderm. Here on the inside is the endoderm forming the gut. And here on the other side here is the mesoderm. And the mesoderm forms muscle, all of the other organs that I've told you about. But this is very limiting. Most organisms in this group have to be very flat because all of their gas exchange and everything has to take place from the outside to the inside. So it's difficult to get gas like oxygen to make its way all the way through. If you were big and thick, it would be very difficult to get the tissues on the inside oxygenated, etc. So they tend to be very flat. They tend to be limited in size, unless they're in very, very specialized environments, as you'll learn in the next class. But most animals have an elaboration on this structure. So these ones here have no body cavity. Here's the next kind. And what these organisms have done, this is typical of a group called the nematodes. They are the roundworms and things like that. They still have three layers. Here they are, the ectoderm. Here's the endoderm, and here's the mesoderm. And you'll see that there's a hollow cavity around the endoderm between the mesoderm and the endoderm. This is called a hemocoel or a pseudocoel. And it's filled with fluid. It may be filled with fluid which conducts oxygen and nutrients and everything back and forth. But its most important function is that it's fluid filled, and fluid is incompressible. And it means that the muscles which are formed in the mesoderm here can act against this fluid filled interior, and the fluid acts as a skeleton, a hydrostatic skeleton. It gives the organism structure, and it allows for specialized movement. So that is its main, but it has a huge disadvantage. And that is whenever you contract this or you do anything, you're going to act on the gut, and the gut has none of its own muscle. It has no way to move material through it. It has to rely on body movement to move stuff through it. It has none of its own musculature. That's a huge disadvantage. Nonetheless, there are, as we go through all of the animal groups, we find that there are a couple of groups that actually do try to exploit this kind of thing. And some of them, the nematodes only have this pseudocoel. But others have a mixture of the two. They have some areas of the body which have this kind of structure. And then they have other areas of the body which have another structure that I'll tell you about now, a true coelom. So in the mollusks, for example, they have a mixture of the two. Now here's what is regarded as being the most advanced of these body cavity structures. And this applies to most of the triploblastic, three-layered animals. They have a body cavity called a coelom, and it is formed by a split in the mesoderm. You can see it here, there on either side. So what this does is it allows for specialization of different kinds of muscle. Here on the outside here are outer muscles. In many animals, it forms skeletal muscles, for example. Here on the inside, there is mesoderm, which can be specialized to form organs, first of all. Many of our organs arise from this inner layer of mesoderm. And it also allows the gut to have its own musculature. So the gut musculature can operate completely independent of all the locomotory musculature and everything else. In some animals, the coelom may also provide a hydrostatic skeleton for the animals as well. That would be true of things like the earthworms, the annelids, and those kinds of things. Mesoderm is extremely versatile, and from the mesoderm tissues can develop many different kinds of tissues and organs, etc., etc., etc. So that is the true coelom. And we need to keep an eye on all of those things. All right? We need to keep an eye on the structure of the coelom, etc., because we're going to need to know where that coelom comes from. There are different ways in which that coelom can develop. And it divides all of the way in which it develops, divides all the animals into two great groups. And these are the protostomes and the deuterostomes. And along with the difference in the origin of the coelom goes a difference in development, in early embryonic development, and a difference in the fate of the first hole that we saw called the blastopore. Now, I strongly suggest that you can use if you printed these out as notes, you can do it here, but I strongly suggest that you start making tables where you list the differences between these two groups, the protostomes and the deuterostomes, because it's tremendously important as we proceed looking through the different animal groups. So the first thing to tell you about is that the protostomes and the deuterostomes have a huge difference in the way in which the zygote develops. In the protostomes, there is a system of cell division which is called spiral. Now, what this means is that you'll see it's somewhat unequal. The division is not just, it's an equal division. But have a look here and you'll see that here is a cell there, which has actually given rise to that cell there. Here is the cell which has given rise to that cell there. These daughter cells are sitting to the side. When cells in protostomes develop in the early, they reproduce in the spiral fashion, and it's called a spiral. Not only that, if I take an early embryo like this in a protostome and I split up these cells, one from the other, separate them, they cannot develop further. They may divide a few times, but they are unable to proceed with development. Their development and their subsequent division is absolutely tied to the fate of their neighbors. This is so that we say that their fate is determinate. There is determinate cleavage. In the deuterostome group, this is completely different. Instead of the spiral divisions, we have instead this, what is called radial division. This cell divides and it produces a daughter cell there, which is directly connected to the original cells. Okay, so both of these cells have arisen from one cell and they remain as closely associated with one another. In the deuterostomes, up to a certain stage, about 16 cells or so in some animals, 8 cells in others. If I split these cells up, if I take these 8 cells and I separate them one from another, each of them will behave as a separate zygote. They'll start division all over again. They'll start their development all over again. And they'll be able to form 8 different embryos. So we say their fate is not tied at this stage to the fate of their neighbors. So we say that this is indeterminate. This is indeterminate division, cleavage rather. And this is true for us. We are Deuterostomes, and that is true for us. Okay, so the second thing is that the Protostomes and Deuterostomes have a different origin for the Mesoderm. So in Protostomes, this is a bit of an oversimplification, but in any case, we'll go with it. The Mesoderm development begins very, very early in both of them. Here is the gastrulation has proceeded, and here we have the arc enteron here. This is endoderm there, and here is the blastopore. And in the Protostomes, the Mesoderm develops as a solid mass of tissue, which arises from cells down here. So we get a clump of cells, and these cells split apart from one another, and this moves up here like this and up here like this until they unite. And you realize that what you then have is a double layer of mesoderm there, around there, with a hollow in the middle, which is the coelom, the true coelom. So that is the protostome development. Deuterostome development, the mesoderm develops from the top here, and this actually folds over, and the coelom is formed from folds which originated from the arc enteron. So it really is like an elaboration of the arc enteron that comes around here and then buds off to form this enclosed coelom around here. So those are the two different kinds of both of them now. When this is complete, both of them have these double layers of mesoderm around here with the coelom in the middle, and both of them at this stage still have one opening, which is the blastopore. And now there is a difference in the fate of the blastopore. And it is this: when in the protostomes, here is the blastopore here, and I just need to move something again. Sorry. Sorry. Zoom, I don't know if you can see them, but Zoom throws up all sorts of things which cover things, and I never know whether you can see them or not. Okay, so here's protostome development. We're going to follow the fate of the blastopore. Here's the blastopore, and in the protostomes, that forms the mouth. And the anus is formed as a separate opening up here. In the deuterostomes, it's the opposite. In the deuterostomes, the anus forms from the blastopore, and the mouth is separately formed. This is a hugely important difference. It looks, doesn't it, as if the protostomes are a mirror image of the deuterostomes. In fact, that's true in many, many ways. In many ways, that is true. And there's a reflection of this in the fact that when we look at the protostomes, all of the protostomes, for example, have a nervous system. Don't forget that the nervous system is developing as a result of elaborations of all of these embryonic tissues. The nervous system develops on the ventral side, whereas in the deuterostomes, think of us, our nervous system is on the dorsal side. And the same thing applies to many, many aspects of the development. They seem to be inversions of one another. And this is very complicated, but it is an actual fact that it arises because the Hox genes that I told you about earlier actually direct these processes differently in the protostomes and in the deuterostomes. So what you need to remember for the moment is that in the protostomes, mouth develops from the blastopore, anus develops separately. In the deuterostomes, the anus develops from the blastopore, and the mouth develops separately to produce the complete gut. Okay, so now let's have a look at the actual phylogeny of these groups. And the phylogeny, of course, I'm afraid, is somewhat complicated. The reason being, this is a very, very diverse group, and we're trying now to put them together with two major inputs of information. The first is morphology, just as they are, body plans, etc., especially their embryology. These are very, very significant. Their similarities and differences in embryology are extremely significant. And lastly, this whole phylogeny has been radically influenced by our studies on DNA similarities, on DNA comparisons of DNA sequence. And these are the groups which we have arrived at. First of all, we realize that there is a common ancestor to all of these. And these groups, all the way from the simplest, which are the peripheral, through all of the rest, are called the Metazoa, the truly multicellular animals. Okay, that's what it means. But there is, we put the peripheral off as a basal group because they don't have true tissues. Those with true tissues, we call the Eumetazoa. And there is there was a Eumetazoan ancestor approximately 680 million years ago, before the Ediacarans. And it's from those roots have arisen all of the other animal groups that we see here. So the vast majority, if you just look through here, please ignore this one for the moment, ignore its symmetry. This is a specialized symmetry. All of these groups have one outstanding feature, and that is that they are bilaterally symmetrical. There are only a few other groups which are radially symmetrical. They're successful groups, but they don't have the huge diversity that the bilaterally symmetrical groups have. So these two groups here, Ctenophora, which I'm not going, they are a jelly, but they're not closely related to the Cnidaria. And the Cnidaria, which are the jellies, Hydra, those sort of things, these are our truly radially symmetrical groups. But their radial symmetry is not a derived character here. Okay, it's not an ancestral character to both of them. These are not sister groups. They're quite separate. All of these we call, all of the rest we call the bilaterally symmetrical animals. And these are predominantly invertebrates. The vast majority of animals are invertebrates. They're all in, this whole group is the Bilateria. They are bilaterally symmetrical. This group, as I told you, the Echinodermata, closely related to us. Their radial symmetry is secondary. It's a specialization that has arisen in them. It's not true radial symmetry. The only group which has a backbone, is not invertebrate, is in this group here, the Chordata, but not all, all of the Chordata are vertebrates. Only one group in the Chordata are vertebrates, and that is us. Here's our group here. This group here, we'll spend our attention to this, the Chordates, the Chordata, and the Echinodermata. These are the Deuterostomes, and they are a clade. The Deuterostomes are a single clade. The Protostomes are divided into two clades, and those are these, the Lophotrochozoa, which are all of these, and then this group here, which are the Ecdysozoa. And we will discuss the characteristics of these in more detail now and when we discuss the invertebrates. So here are this group called these groups called the Ecdysozoa and the Lophotrochozoa. Now these are all invertebrates. The ecdysozoa are characterized by having an external skeleton. To remember this, realize that the insects and the crayfish and all of the arthropods, those are all ecdysozoa. Ecdysing means shedding your skin. The ecdysozoa are those which have to shed their skins. They have a rigid outer covering which needs to be shed so that the animal can grow. And there are two groups which are in the ecdysozoa. They are the arthropods and the nematodes, the worms. I know that it seems very strange, but the single derived character that ties them together is this outer chitinous covering, the exoskeleton. Very thin in the nematodes, but very thick and strong in the arthropods. The other giant, let me just go back. They're the Lophotrochozoa. Okay, they are forming a single clade there. That's our next group. Here are the ecdysozoa, the nematode and the arthropod. Here are the Lophotrochozoa. Very, very diverse, and in fact, they're quite a contentious grouping. Some people disagree with it and don't think it's a single clade. But in any case, we will regard it as a clade. And there's one obscure group in there called the ectoprocs, which we're not going to talk about. And then a whole slew of other very diverse organisms, such as the annelids and the mollusks and all of these. They're morphologically very, very different, but it is clear from molecular studies that the Lophotrochozoa are very closely related, despite all of their morphological diversity, and despite the fact that they have diverse larval stages and all the rest of it. This type of larva, the trochophore larva, occurs quite frequently through the group. So, for example, mollusks and annelids may have trochophore-like larvae. But it's their molecular structures, it's their DNA similarity, which really define this group. So remember the ecdysozoa and the Lophotrochozoa. Those are the two protostomial groups, but they're not grouped as the protostomes. Okay, they are both protostomial, but they are separate from one another. So the next group that we need to talk about, of course, are the Deuterostomes, the Deuterostomia, and that is our group. Again, it's a diverse group, not nearly as diverse as the Lophotrochozoa, but it's a group which includes invertebrate forms and then the vertebrates as well. And we have close invertebrate relatives. Going to Thanksgiving, you may think that that is particularly true, but nonetheless, believe it or not, all of these groups shown here are all related. These are all chordates. And the morphological feature which ties all of the chordates together is that at one stage or another within the life, often in the larval form, but in many of them in the adult form, it persists into the adult form as well. There is a particular structure, a gelatinous rod, cartilaginous rod, I would say, which runs down the dorsal side of the organism, which is called the cord, the notochord. I'm sorry, my brain went dead from it. It's called the notochord, and that notochord is a feature of locomotion, of particularly of larval forms. But then in some of the chordates, it persists into the adult as well. We have a notochord in our embryonic stages, and it persists. It gets turned into bone later on and forms part of the vertebrae. So all of these are our relatives. This very strange group of wormy-looking things called the hemichordates. These are echinoderms, which are kissing cousins to the chordates. And then these here are all chordates, vertebrates like us. Then these invertebrate chordates as well. And these are even closer to us than the echinoderms. So that should kind of humble us a little bit. Okay, so in our next class, we're going to move on to start discussing invertebrates, and we'll return to many of these groups. So please make sure that you go over these different groupings, go through those phylogenies, and make sure that you understand them. And then I will see you on Monday.